Good morning, students. Today we are going to discuss about more opposed fertilization structures and events, Apomicious and Poli Embroidery from Chapter 2, Sexual Reproduction in Flowering Plants. Let us go to structures and events from post-fertilization, that is seeds, formation of seed. Seed is the final products of sexual reproduction in angiosperms or in flowering plants. And seeds are often described as fertilized ovule, and this ovule is formed inside the seed, inside the ovary. Generally, seed consists of seed coat or seed coats, cotyledon or cotyledons, and an embryo axis. Seed coat is the outer tough protecting coat. This is the tough protecting coat, and this is what we call seed coat. Cotyledons are the thick and swollen simple structures, and they are often referred to as seed leaf. Embryo axis are the structures of embryo that has embryonic shoot and embryonic root. Seeds may be non-albuminous or ex-albuminous. Non-albuminous seeds are those seeds which has no residuals of endosperm, as it is completely consumed during embryo development. Example, pea groundnut. In seeds of pea and groundnut, there is no endosperm left behind, as it is completely eaten up by developing embryo. Ex-albuminous seeds are those seeds that has some amount of endosperm left on the seed, as it is not completely eaten up by developing embryo. Example, wheat, maize. Integuments of ovules harden as protective seed coats. If you look at the picture, we have ovary, and inside the ovary we have ovule. If you look at the ovule, there are two covering of ovule called integuments. And when the ovule turns into seed, these integuments will turn into protective seed coats. The micropyle remain as small spores. Look at the micropyle here, small opening in the ovule. These micropyle continue to remain as small pores in the seed coat, and these will facilitate the entry of oxygens and water into the seed during germination. As the seed matures, the water contained of the seed reduce and seeds become dry. The moisture content reduced to only 10 to 15% by mass. Even the metabolic activity of embryo slow down, and the seed enter a state of inactivity called dormancy. And during dormancy, the seeds remain inactive, but they become active and germinate if favorable conditions are available. What are the favorable conditions? Like available of suitable temperature, humidity, water, air or oxygens, etc. As ovules mature into seeds, the ovary develops into fruits. The ovary develops into fruits, and the wall of ovary develops into wall of fruits called pericarp. Okay, as the ovule turn into seeds, simultaneously the ovary also turn into fruits, and the ovary wall turns into wall of fruit called pericarp. If the thalamus contributes to fruit formation, such fruits are called fall fruits. Thalamus are referring to receptacle where the different parts of flower are born. So if the thalamus contributes to fruit formation, then such fruits are called false fruit. Example, apple. If only ovary develop into fruits, then such fruits are called true fruits. Example, we have mango. Although fruits are results of fertilization, that means fruits are a result of fertilization in most of the species, there are few species in which fruits develop without any fertilization. So such fruits are called parthenocarpic fruits. Example, banana. Now parthenocarpy can be induced. That means we can produce parthenocarpic fruits through the applications of growth hormones, and such fruits are seedless. Now let us go to viability of seed. Viability of seed means the capacity of the fruit of the seed to carry out its functions like germination and grow up into new plant. How long do the seeds remain alive after they are dispersed, or how do the seeds remain alive once they are detached from mother's plant? In few species, the seeds lose viability within few months. But some seeds of large numbers of species can live for several years, can remain alive for several years after they separated from mother's plant. Some seeds can remain alive for even hundreds of years. Let us see some example. Lupinus articus, excavated from Arctic tundra, germinated and flowered after an estimated record of 10,000 years of dormancy. The seeds of Lupinus articus remained dormant for 10,000 years, and after 10,000 years of dormancy, the seed germinated and flowered. A recent record of 2,000 years old viable seeds is of the dead palm discovered during the archaeological excavation at King Herod's Palace near the Dead Sea. So this is the Lupinus articus flower, and this is the Lupinus articus seeds that can remain dormant for 10,000 years. And below is the plant of dead palm, and this is the dead palm seeds that remained dormant for 2,000 years. Now let us go to next topic, that is Apomicious and Poli Embroidery. What is Apomicious? Apomicious is the mechanism of seed production, formations of seed without involving the process of meiosis and fertilization. In what happened in sexual reproduction, meiosis take place and fertilization take place, then only a seed is formed. But in case of apomicious, seed is formed like in case of sexual type of reproduction, but here they bypass meiosis and fertilization process. So apomicious is a form of asexual reproduction that mimics or copy sexual reproduction. They are actually asexual reproduction because they don't involve fertilization, but they copy sexual reproduction, type of reproduction by producing seed. What is the importance of apomicious? It plays an important role in hybrid seed production. Okay? It allows commercial use of hybrid seeds without conducting hybridization again and again. Now the problem of hybrid seeds for farmer is that the hybrid seed showed in the second years usually lost the trait of hybrid in the progeny, and we all know that hybrid seeds are quite expensive, so some farmer cannot afford hybrid seed. But if we produce this hybrid seed through the process of apomicious, then the hybrid seed can continue to use for many generations. The genetic nature plant produced by apomictic embryos is same as the parents. Okay, so it is a form of asexual reproduction. Therefore, the plant produced through this apomictic by this apomictic embryo will be exactly the same as the parent, so they can be referred to as clone. Apomicious can be seen in some species of asteraceae and grasses. Now let us go to Poli Embroidery. Poli stands for many. It is the formations of more than one embryo in the seed. Okay. Actually, A single seed is supposed to have only one embryo. Okay? An ovule has only one embryo sac, so when the ovule turns into seed, the seed is supposed to have one embryo. But sometimes there is a presence of more than one embryo in a single seed. So that is what we call Poli Embroidery. This phenomenon was initially discovered by Leeuwenhoek in 2019 when the seed in citrus have two germinating embryos. Citrus, pinus, orchid, mangoes plant usually have this phenomenon. What could be the reason for polyembryony? Sometimes the antipodal cells, okay, there are 3 antipodal cells, 2 synergids and nucellus. All these are also give rise to embryo. That is the reason for the presence of more than one embryo in the single seed. So that's all for today. Thanks for watching.